Good health is a precious thing worth fighting for. Tragically, infectious and other diseases present an increasing threat to all of us. The development of new, innovative medicines can help us face these threats. When we talk about developing cutting-edge therapeutics, it is important to consider more than 30 years of biotechnology evolution. The initial success of biotechnology has been driven by the ability to put DNA into living cells and make them produce medicines on a large scale. The manufacturing of insulin is the first example of that. This was first done in the 1970s by introducing the human insulin gene into bacteria. These bacteria then produced insulin, which is now used to successfully combat diabetes. As the industry became interested in the production of more complex medicines, more sophisticated cell types were required as a production platform. An important breakthrough came in the early 1980s when mammalian cells could be used for large-scale manufacturing. To date, animal cells such as rodent cell lines are used extensively for the production of biopharmaceuticals, including blockbuster drugs. But in the 1990s, Crucell recognized the need for a new human cell line that was specifically developed with pharmaceutical manufacturing in mind.
It really was unique. For the first time in history, a human cell line was generated with the intent to manufacture biopharmaceutical products. When I started Introgene, the predecessor of Crucell, we had the need to manufacture products on cell lines. We were focusing on gene therapy, and for that we required human cells. In order to make a human cell line, I realized the relationship that we had with Professor Van der Ep at Leiden University was crucial, because he had identified the way to use the E1 gene to immortalize cell lines, and that's how the PERSY-6 project started.
To make a long story short, after creating a predecessor cell line, I worked closely together with Dinko Valerio and succeeded in developing the PERSY-6 cell line under defined conditions. This meant that every aspect of the process was clearly documented, providing a complete history of the cell line.
The PERSY-6 cell line is made from a single human cell that has been immortalized. The cell can keep dividing and replicating indefinitely. This was achieved by inserting the so-called E1 gene from a commonly occurring adenovirus. This altered the cell's genetic properties and allows it to replicate itself into an endless series of identical copies. Even though the PERSY-6 cell line was developed for making gene therapy products, it was soon found to have a much broader range of use.
We found out that many human viruses grew extremely well on PERSY-6, allowing mass manufacturing of vaccines based on those viruses. We could do that reliably, safely, easily, and at low cost.
Viruses can be introduced into the cells, grown to large numbers, and then inactivated or killed to make vaccines. Using this type of process, Crucell is able to develop vaccines against the likes of influenza and West Nile. But it was also found that the PERSY-6 cell line could be used to make recombinant vaccines. Effective vaccines against severe infectious diseases such as AIDS, Ebola, or malaria require new types of vaccination strategies because the traditional technologies are not effective or safe. PERSY-6 is very well suited for the production of recombinant vaccines, where a gene for disease-causing virus or parasite is inserted into a vehicle called a vector. This vector, when injected into the patient, helps the body to create an immune response against the virus or parasite. In addition to vaccine production, the PERSY-6 cell line offers improved safety, scale, and yield for the protein market, a multi-billion-dollar industry. In fact, the PERSY-6 production platform has been optimized for therapeutic protein and monoclonal antibody production on a commercial scale.
We have succeeded in culturing the PERSY-6 cells to a density of 150 million cells per milliliter, and this is unique in the industry. At this cell density, about half of the volume of the culture medium is occupied by cells. The PERCY6 cells also successfully performed at the 20,000 liter scale, and this is the maximum size reactor available for mammalian cell culture. Imagine the production capacity of a 20,000 liter bioreactor, which is about the size of a swimming pool.
Based on these successes, Crucell, together with manufacturing and technology partner DSM Biologics, is taking protein production to the next level. The two companies have joined forces to create the Persivia PERCY6 Development Center in Cambridge, Massachusetts. Persivia will further develop the PERCY6 cell line and will also transfer existing and newly developed PERCY6 expertise to licensees. The end goal is to lower the production costs of therapeutic proteins so that more patients can benefit from these medicines.
The development of novel biotech drugs like antibodies and proteins for unmet medical needs is a lengthy and cumbersome endeavor. With bundling our forces here in Boston, Persivia will serve its clients better to improve speed to market. We will continue to increase our efforts to bring innovation to global health.